Hello, my name is Talia Biondo. Today is June 21st, 2026, and we will be talking about Wilson's disease, which is a disease of cellular copper transport. Before we dive into the details, let's take a look at our overview. Wilson's disease is an autosomal recessive genetic disorder that causes copper to accumulate in the body. Copper is needed for normal cellular function, but excess copper becomes toxic to cells and tissues. Because copper can no longer be regulated and eliminated properly, it gradually builds up in organs such as the liver, brain, and cornea. The liver is usually affected first because it plays a central role in copper metabolism and excretion. Over time, copper accumulation causes cellular injury, organ dysfunction, and hepatic, neurological, or psychiatric symptoms. Let's dive into how and why this happens. Wilson's disease is caused by inherited mutations in the ATP7B gene, which, as you can see in our photo, is located on chromosome 13. ATP7B contains the instructions for producing the ATP7B copper transport protein, which is found primarily in hepatocytes or liver cells. When ATP7B is mutated, the protein becomes unstable, it localizes incorrectly, and the copper transport activity is destroyed. More than 600 ATP7B mutations have been identified, and these mutations can differ, but they all affect copper regulation. Wilson's disease follows an autosomal recessive inheritance pattern, meaning affected individuals inherit disease-causing ATP7B variants from both parents. The inherited mutation is the starting point of this whole disease process. So let's take a look at what it seems like when we actually process copper correctly and normally. Normally, copper enters hepatocytes and is transferred to ATP7B by the copper chaperone ATOX1. ATP7B performs two essential functions. First, it incorporates copper into ceruloplasmin, which is the primary copper-carrying protein in the blood. Second, it transports excess copper into bile, which serves as the body's primary copper excretion pathway. Together, these processes maintain copper homeostasis by allowing copper to be transported, used, and eliminated in a controlled way while preventing toxic accumulation. Now let's take a look at the failed metabolic pathway of Wilson's disease. On the left, you can see the normal copper metabolism. On the right side, this is the mutation, or one of them. So, when ATP7B becomes dysfunctional, normal copper regulation fails. Copper can no longer be incorporated efficiently into ceruloplasmin, and excess copper can no longer be removed effectively through bile. Because copper continues entering the body but cannot be eliminated normally, it begins accumulating more and more and more inside the hepatocytes. Over time, copper homeostasis is lost, and instead of maintaining a balance between copper uptake and copper elimination, the cell stores increasing amounts of copper, which initiates cellular injury and tissue damage. Let's take a look at its impact on cellular functions. Here you can see a hepatocellular injury model of Wilson's disease. This is a liver cell, a demonstration of the failed metabolic pathway due to Wilson's disease. So, as copper accumulates inside the hepatocytes, several important cellular functions become disrupted. Copper overload can cause protein malfunction, lipid peroxidation, DNA instability, and impaired intracellular copper trafficking. Copper can accumulate within the cytosol and nucleus, where it interferes with normal cellular activity and genetic stability. ATP7B dysfunction also disrupts normal protein localization within the trans-Golgi network, which further interferes with copper handling. As these abnormalities accumulate, the cell gradually loses its ability to maintain normal structure and function. How can this excess copper damage cells? While the direct cellular damage is caused by the toxicity of the copper, excess copper promotes oxidative stress and the production of reactive oxygen species. These reactive molecules damage proteins, lipids, mitochondria, and DNA, leading to progressive cellular injury. As damage continues, hepatocyte degeneration, inflammation, and tissue remodeling develop. Over time, this combination of oxidative stress and inflammation impairs normal liver structure and function. How does this snowball into a bigger picture? Here we can see a demonstration of all the symptoms and signs of Wilson's disease. So, although Wilson's disease begins at the cellular level, its effect eventually spreads throughout multiple organs and body systems. Copper accumulates first in the liver, where it can cause hepatitis, cirrhosis, and liver failure. As copper continues to build up, it can also affect the brain, the basal ganglia, and other tissues, and the cornea, specifically. Let's remember that one. Neurological symptoms may include tremors, movement disorders, impaired coordination, impaired motor control, and features of Parkinson's. Psychiatric symptoms may include depression, irritability, personality changes, cognitive impairment, and even psychosis. As a result, Wilson's disease affects liver function, neurological regulation, movement control, cognition, emotional regulation, and behavior. If left untreated, severe liver or neurological complications can become life-threatening. So this is a very serious disease. The signs must be taken seriously. Let's take a look further into the genetics, inheritance, and risk factors of Wilson's disease. So because ATP7B mutations can be passed to future generations, genetic testing is important for diagnosis and family screening. Wilson disease follows an autosomal recessive inheritance pattern, like we said, which means affected individuals inherit one disease-causing ATP7B variant from each parent. Genetic sequencing can identify specific disease-causing variants directly, because this is very well studied. Genetic testing can help confirm a diagnosis. It can identify affected relatives, support family screening, and provide prognostic information. Some individuals have even been identified through family screening before symptoms have developed, which seemingly is the best method. Although ATP7B mutations are the primary cause of Wilson disease, genetics alone does not fully explain disease severity. Environmental influences, dietary factors, modifier genes, and epigenetic factors may contribute to differences in disease presentation. Wilson's disease is not caused by cell cycle checkpoint failure. Instead, it develops because, as we said, the ATP7B mutation disrupts copper transport and copper metabolism. So how is it diagnosed other than that? Wilson's disease is diagnosed using a combination of clinical findings, lab testing, ophthalmologic examinations, and genetic analysis. Common laboratory tests include serum ceruloplasmin measurements, 24-hour urinary copper testing, liver copper measurements, and ATP7B genetic testing. Ceruloplasmin is actually particularly important because ATP7B dysfunction often results in reduced levels. Physicians may also look for the, I shouldn't say iconic, but the Kaiser-Fleischer rings, which are significantly known. They develop as when copper accumulates in the cornea, and they're considered a classic sign of Wilson's disease. This is what people think of when they're thinking about Wilson's and want to identify it on the left. Anyways. So because no single test confirms every case, many clinicians use the Leipzig scoring system, which combines clinical findings, lab results, eye examination findings, and genetic information to establish a diagnosis. The most common treatment approach is chelation therapy. Established treatment focuses on lowering copper burden and preventing additional copper accumulation. That's the standard right now. So D-penicillamine and trintane bind excess copper and promote urinary copper excretion. and they help remove accumulated copper from the body. Those are oral pills. Sorry, I'm a little nervous. Another established treatment is zinc therapy. Instead of removing stored copper, zinc reduces intestinal copper absorption and helps prevent additional copper accumulation. Because underlying ATP7B mutation remains present, treatment is generally lifelong, and unfortunately, in severe cases involving advanced liver disease or liver failure, then liver transplant may be necessary. A transplanted liver contains functional ATP7B protein and can restore more normal copper metabolism. When treatment begins early, many patients can achieve long-term disease control and significantly improved outcomes. Alternatively, the current research and the future outlook is focused more on addressing the underlying genetic cause of Wilson rather than treating it after the fact. So, although right now we're not directly treating the underlying ATP7B defect, that's what they're gearing towards here. So, one promising area of research involves hepatocyte-targeted lipid nanoparticles that deliver functional ATP7B messenger RNA directly into liver cells. The goal is to allow liver cells to produce functional ATP7B protein and restore normal copper transport. In the Wilson disease mouse models, ATP7B mRNA therapy reduced hepatic copper accumulation, restored copper homeostasis, improved ceruloplasmin activity, and improved copper regulation across multiple organs. Researchers are also investigating mutation-specific therapies because some ATP7B variants retain partial copper transport activity. Additional research focuses on improved biomarkers and newborn screening approaches that may allow earlier diagnosis and treatment. The outlook for Wilson's disease is very encouraging because future targeted therapies address the underlying mechanism, as I said, rather than just managing symptoms for your whole life. So let's wrap it up. How can allied practitioners help? Dietitians, physical therapists, occupational therapists, mental health professionals, genetic counselors, everyone gets together to tackle this using the treatment methods that I just listed, but these are the allied practitioners that partake in that process. And then let's just remember as we say goodbye that Wilson disease is caused by an inherited ATP7B mutation that disrupts copper homeostasis. And thank you so much for watching. Have a great night.